Good morning to all. Uh, Today, the Department of Botany and Internal Quality Assurance Cell, Dadapatil Rajale Arts, Science and Commerce College, Adinathnagar, Taluka Pathardi District, Ahmednagar, Maharashtra, is organizing one-day national webinar on COVID-19 basic understanding and aerobiological perspective. So, we have with us Dr. K. Ramchander Rao as a resource person. Here, the principal, Honorable Principal Dr. Temkar sir, faculty in charge of arts, Dr. M.S. Tamboli, IQAC coordinator, Professor R.T. Gholap, then Dr. S.R. Bharate, Department of Botany, all my colleagues and all the participants. I welcome you on behalf of the Department of Botany and Internal Quality Assurance Cell, Dadapatil Rajale Arts, Science and Commerce College, Adinathnagar, Taluka Pathardi, District Ahmednagar, Maharashtra State. Here, I request in charge principal Dr. M.S. Tamboli to introduce briefly about the today's webinar and about the college. Over to Dr. M.S. Tamboli.
Webinar, Dr. K.S. Ramchander Rao, Consultant and Director, Aerobiology, Muscat. The faculty of the Department of Botany, Dr. J. Nehul and Dr. S.R. Bharate, all my colleagues and dear participants. This is a good initiative taken by the Department of Botany by organizing a national webinar on the topic COVID-19 basic understanding and aerobiological perspectives. On behalf of the management, our principal Dr. R.J. Temkar, NAC coordinator Professor R.T. Gholap, and all faculty members and students, I heartily welcome Dr. Rao and all the participants. I take this opportunity to inform about Dadapatil Rajale Arts, Science and Commerce College, which is located in a rural and remote area. But despite all adverse conditions, our teachers are giving their best by taking several initiatives. It is our sincere effort to give our best for the overall development of this institute. The management and the principal, they are very much supportive and encourage all the teachers to undertake various academic activities. Soon we are going through NAG for the third cycle, and we are very much confident to secure excellent grades in this third cycle. Our staff is making sincere effort to staff is committed for the overall development of the institute. We are making extensive use of ICT for teaching, learning and administrative works. And we are very much prepared to adopt the new educational policy which is introduced by the government of India. And I am very much confident that in this process, our institute is going to be as one of the leading institutes located in a rural area. I appreciate the Department of Botany for organizing today's webinar and inviting such a distinguished person like Dr. Rao, who is all the way here from Muscat. I must say that the Department of Botany is one of the leading departments in our institute, and both the faculty are very active. I'm sure that today's webinar will give a new insight to all the participants. Once again, I welcome Dr. Rao sir and all the participants, and I conclude my speech. Thank you.
Thank you, Dr. About the college, the department, and about the today's webinar. So here, before starting the webinar, I would like to give an introduction of today's speaker and resource person. So today we have with us Dr. K.S. Ramchander Rao. Dr. Rao has completed his degree from Osmania University, Hyderabad, and PG and PhD degree from Marathwada University, Aurangabad. So presently he is working as a professor, Applied Sciences, Department, Higher College of Technology, University of Technology and Applied Sciences, Al Khuwair, Muscat, in Sultanate of Omen. Then he is a consultant Aerobiology and director, Abu Hussein United LLC, Muscat, Sultanate of Oman. Then he is PhD FIAS. He has more than 35 years of teaching experience. His areas of research interests are immunology, aerobiology, allergology, occupational health disorders, environmental biotechnology, nano-aerobiology, nanotechnology, and indoor and outdoor air quality. So he has taught various subjects at postgraduate level like fundamentals of biology, genetics, cytogenetics, molecular biology, microbiology, bioinformatics, evolutionary biology, cell biology, plant biotechnology, environmental science, genetic engineering, bioprocess engineering, immunology, computational, molecular biology, nanobiotechnology. His research, he has supervised more than 30 students for PhD degree. Then he is a principal investigator of many major research projects funded by DST and ICMR. He has guided 30 BTech and MTech and PhD dissertations. Then he has organized several national and international conferences. Then he has worked as a resource person in different institutes, public enterprises, in Osmania University campus, then PS Telugu University Hyderabad, All India Radio. He was also a coordinator of IGNO Study Center, participated in radio counseling sessions. Then he has delivered more than 30 invited lectures in international and national conferences. He is a fellow of Indian Aerobiological Society for more than 35 years. Then several research papers, they are to his credits. Then near about 60 papers have been published by Dr. K.S. Ramchander Rao in the leading international and national journals, both in India and abroad. Then he has visited different countries like Sweden, Germany, USA. Then he was a visiting scholar for 1 year, that is 2005 to 6, in the Department of Health Medical School, University of Cincinnati, USA. Then he is a life member of Indian Aerobiological Society. then Institute of Botany. He is a member of International Association for Aerobiology, Phycological Society. Then he is a working or executive committee member of Vasundhara, the Earth, member of Pan America Association for Aerobiology, member of Environmental Society for Omen. So today we have such an eminent scholar and resource person, Dr. K.H. Ramchander Rao. So I am sure that we will be benefited by his talk. And again, I welcome all the participants, my principal coordinator, Dr. M.S. Tambodi, then Dr. Bharate, and all the participants on behalf of the Department of Botany and IQAC cell, Dadapatil Rajale College, Adinathnagar, Maharashtra. Again, I welcome and I invite Dr. K. Ramchander Rao for his lecture. So I request Dr. Rao to start his lecture on COVID-19, basic understanding and aerobiological perspectives. So thank you to all, and I over to Dr. K. Ramchander Rao. Thank you.
Thank you, Dr. Nehul, for introducing me. Dr. Nehul happens to be my student during his post-graduation in Chalisgaon. And I also thank Professor Temkar, principal of this college, Professor Bharate, Department of Botany, Professor Gholap, Co-ordinator IQAC, and also Dr. Tamboli, in charge principal, who has introduced just a while ago about the college. So I thank all Nehul and his team for inviting me to give this talk on the current burning issue, that is COVID-19. Now my talk, I straight away go into the lecture on COVID-19, as has been explained by Dr. Nehul. That the title of my talk will be Basic Understanding and Aerobiological Perspective. I hope I am audible. Am I audible to all of you? Hello. Am I audible to all of you? Yeah. So let me proceed now. At the outset, I would like to introduce myself. That already introduction has been given. I'm right now. I'm living in Muscat, and working as consultant in aerobiology with a firm after my recent retirement from the service in University of Technology and Applied Sciences. Now, before I further proceed into the lecture, I would like to pay my condolences, my respectful regards to the late Professor S.T. Tilak, who has been my mentor, who has been considered aptly called as father of Indian aerobiology, and who has been my PhD guide, who is no more, but his contribution still cherishes in the country and in the international arena. Now, going into the talk, we are now going through a phase where there are terrible problems posed by a virus by name disease called as COVID-19 disease, and the virus called as SARS-CoV-2. So before I throw some light on the SARS coronaviruses, let us try to understand what are the viruses. Most of us are aware of it. Most of us have been studying, like the teachers as well as the students who are attending now this webinar. I believe that they are aware of the viruses, uh, because since they've studied it somewhere at some point of time, either at 10+2 or in the schools or even in the degree college, college, undergraduate college. At some point, they've got some information about the viruses, which have been considered and which have been debated as either as living organisms or as non-living entities. There's a big debate still going on that whether should we consider the viruses as living creatures or should we consider simply the viruses as non-living entities containing the genetic material. In the simple definition part, a virus is a small parasite or a microorganism that is smaller than a bacterium that cannot grow or reproduce by itself. So we know that, when we have been studying on the viruses, that we are aware that the viruses cannot replicate by themselves without the help of the host. The host could be any other living organism. So therefore, they are, we can consider in our scientific terminology, can be called as obligate parasites. They require a living host compulsorily, and they invade living cells and use their chemical machinery to produce more viruses. That is what is called as growth and replication. And interestingly, viruses may contain either DNA or RNA as their genetic material. Now, when there are different ways that viruses can be classified. We don't want to go into all those details of the classifications, but at least the simple definition, simple classification, when we see that based on the host which they infect, the viruses can be classified as the plant viruses, the animal viruses, the human viruses, the bacterial viruses, which are popularly known as the bacteriophages, the fungal viruses, mycoviruses or mycovirology, the branch called as algal viruses, the viruses which infect the algal cells, or sometimes they are able to infect the cyanobacteria, and they are popularly called as cyanophages. So this is how the viruses have been classified based on the host. This is not a general classification as being given in the Baltimore system of classification, but this is how we have convention. For our convention, we have divided them into the viruses which infect plants are being kept under plant viruses, and we are fortunate enough to say that there is no transfer occurring between the plant viruses and the animal viruses. But there is a transfer occurring between the animal and human viruses because we belong to the animal kingdom. There are bacterial viruses, bacteriophages, which have laid the foundation, the microbiology. When we studied the sexual transformations, which sexual recombinations which occur in the bacteria, we call them by name as transformation, transduction, and such things, conjugation, when it occurs between the two bacterial strains. So the fungal viruses, we have many varieties of fungi where the viruses are able to cause infection to them as well. So ultimately, they require their survival. They want their survival viruses, and in order to achieve that, they can enter into any kind of host, a living host. And surprisingly, we see there are so many varieties. They are omnipresent. They are present in water. They are present in soil. They are present in air as well. Now, the topic which will be debated in the next course of slides. Now, going back into the history or the scientific theories which have been proposed, scientific observations which have been made, that viruses as precursors of cellular life. How, when it comes to the origin of the viruses, how they originated, how they came into existence on this planet Earth. Where do they survive? How do they survive? Outside the host, do they survive? If they survive outside the host, how long they will survive? So for this, there were earlier interpretations which were made as viruses as precursors of cellular life. So one belief, one school of thought, believes that the viruses would have been the, precursors to the cellular life. That means, the origin of life itself might have been with the viruses. When we believe that, as per the Darwin's theory of evolution, and as per the origin of life, we believe that the RNA was the precursor for a cell. And if we believe so, and we give credence to that particular interpretation or the theory, then we have to believe that the viruses could be the precursors of cellular life, or when the cell started functioning on this planet. So rather than being derived from existing cells or cellular genetic elements, viruses may have existed before cellular life, which are self-replicating units in the ancient virus sphere, may have gained the ability to form membranes and cell walls, leading to evolution of the three domains of life. So viruses then continue to evolve with the evolving hosts. So this is one interpretation which has been given credence as the viruses being believed as the precursors of cellular life. That means even before the bacteria came into existence. So the second way of thinking is that viruses originated either prior to or from cells. So one which we considered earlier is that pre-cellular, that is before cells have come into existence, as the precursors. Now here the cells might have evolved early, and from those cells a pre-cellular scenario is incompatible with the proposed generic definition of virus propagation inside the cells. Because if we have to believe that the viruses have originated before the cells got formed, then the credence given to that theory becomes invalid, as we know that the viruses always inhabit the cells. So in turn, the origin of viruses has been attributed to archaeoviruses from archaea, bacterioviruses from bacteria, and eukaryotic viruses from eukarya. We know that we have gone from the five kingdom system of classification as given by Whittaker to the three domain system of classification, where we consider three domains of life. Those are archaea, prokarya, and eukarya. So this is how three domains have been considered, as you can see here in this slide. The E, B, A, A standing for archaeoviruses, and then B standing for bacterioviruses, and then E standing for eukaryoviruses. So now, interestingly, that there was a terminology which has been used here in the earlier slide: last universal common ancestor. Now that means this virosphere could have acted as the base, the foundation for the viruses to get evolved into different streams, different varieties, different structures, as we know now. So therefore, as these viruses share several conserved protein folds involved in virion synthesis and other functions, now we have to understand that virions and viruses. So virus is the existing cell or the existing structure which is present in the host, and virions are considered. There is a difference, slight difference between this virus and virions. The virions are the reproductive propagules. Once it is inside the host, then its duty is to replicate. It has to utilize the machinery of the host genome and host cell function to prepare its own structures, its own entities, its own virions. So this is what the difference between the virion and the virus. So this indicates that the modern cells, leuca into modern cells, diversification of leuca into modern cells. So this is as given by Nazir et al. In 2020, who spoke about the LUCA as the ancestral stock. Now, when if we have to understand about the existence of the viruses, as I mentioned in my earlier slide, that they are omnipresent, they are present everywhere. And you may be amazed to understand, to know that nowadays there is a credence also being given to the space as a source of viruses. That means viruses might also have been present, even might be in the International Space Station, and maybe in the outer space, maybe in the other planets as well. We don't know, because there's a lot of work going on the existence of life on the other planets, like Europa, Titan, then Mars, and also Neptune. A lot of work is being carried out, and researches are going on, and especially NASA and is very deeply involved in these researches. And we are also, ISRO is also not behind in trying to explore the unexplored realms in the universe. There are interconversions of viruses and transposons in evolution. There is an exchange. You must know, I think most of you are aware of transposons, the transposable elements, which are part and parcel of every genome, whether it is eukaryotic genome or anywhere, the prokaryotic genome, etc., etc. So these transposons are present in the sometimes we call them as the jumping genes. So these are repeatable elements. They are present. They cause problems also. Because of the presence of these transposons, we do get several diseases. Maybe genetical diseases are also being found because of the presence of these transposons and their expression. The distribution and composition of the transposable elements and viruses are specific for different domains of life, which suggests that interconversions in evolution in which transposable elements are sources of viruses. So that means they are trying to survive, like the human beings, like the animals, like the plants, like all the living organisms, bacteria. All these are, trying to survive, right? So it is essential for them to propagate their life. It is essential for them to survive. They have to, if they have to live, they have to do something. Now this is how the viruses also do the same thing, that in order to replicate, in order to survive, they also have to infect the host cell, and then also they have to replicate in order to increase their population. Thus, DNA viruses constitute a larger portion, as in prokaryotes, and the DNA transposons are also the most common transposable elements. The prevalence of RNA virus is typical for eukaryotes, in the genomes of which the retrotransposable elements are very, very prevalent. That means our genome, the so-called junk DNA as we call, is also comprising of several transposable elements. Part of it might be of the viral genome. That means viral genome is also associated with the human genome for from time immemorial. Just a while ago, I was mentioning about the pathogens which might be coming from the outer space. The pathogens from outer space. This was the book written by Fred Hoyle, and who is no more, late, who has expired in 2001, and then Vikramasinghe from Sri Lanka. So these two authors have written a book called as Diseases from Space, published in 1979, that was authored by these two authors. They propose that many of the most common diseases which afflict humanity, that is such as influenza, common cold, whooping cough, have their origins in extraterrestrial sources. This has been criticized by many. As as in science, anything wants has to be supported by proof, but it has not been completely disapproved, because in science, always there are fictions. Have many of the fictions in the science have become facts now today. So therefore, they believe strongly that the life existed on the other planets, or still might be existing even now, and the life could have originated from the outer space and landed here as the seed material on this planet, and that has given rise to several species, which is, of course, opposing the Darwinian theory of evolution. Already there are so many counter theories which have been proposed, which say that Darwinian theory of evolution might not be 100% correct. But whatever it might be, that there are so many people who have criticized this theory, but just for our understanding, whether space contains viruses, and recent investigations have revealed that viruses do present in the outer space. They think, many scientists thought that, they might not be present, they may not survive, when they are present under that such pressure and then zero gravity situation. But the reality is that even International Space Station, I think the scientists who are astronauts who are staying there, they are, they have come out with the result that the viruses are there in association. Bacteria have been isolated, so that means the viruses could also be there with the plant material or animal material, or the human beings themselves. They might have carried the viruses, or even with the bacteria which have crossed the domain and then have entered into the outer space along with the human beings, might also be having the viruses. This is what the discipline called as astrobiology, formerly known as exobiology, is an interesting interdisciplinary scientific field that studies the origins, early evolution, distribution, and future life in the universe. So, astrobiology considers the question of whether extraterrestrial life exists, and if it does, how humans can detect it. And well, of course, now lot many researches are going on Mars, planet Mars, that and several people are believing, the scientists have come out with the results of the investigation that the life existed in the past on Mars. Now, even in the future, in the coming years, there are going to be some people who are being sent onto the planet Mars, and they are undergoing the training, how to survive. That will be one-way journey. Mars One is doing all these things, and the people will be staying there, and they won't be returning back. And for which, of course, they have committed themselves that they don't mind that they lose their life there on the planet Mars. But this is how the human intelligence is going to explore into the other planets in the universe, whether the life is existing or existed in the past, or in the future, whether life it is possible. Are viruses the new frontiers of astrobiology? Viruses are the most abundant form of life on Earth, but viruses are their seed-like dormant state known as virions, are outliers in our search for life on other planets. But interesting thing happened very recently. Viruses are mentioned 6 times in NASA's 250-page long current astrobiology strategy. Imagine, NASA itself has mentioned it 6 times in the 250-page report. Right, the authors of a recent paper which published by Berliner, Tomihoro, and Stedman. These people have published this paper in 2018. The title of the paper is Astrobiology: Viruses at Large in the Universe. They believe that viruses are present. It is a review paper. They believe that viruses are present in the universe. So therefore, there is a need to redefine the viruses. The ongoing COVID-19 pandemic, most of us are going through it now. It is not over, and when it will end, nobody knows. And where is the full stop? Nobody knows. Or we should develop the herd immunity, maybe a possibility, or should we go for the vaccines? And the vaccines are the solution, yes, of course. So all these possibilities are existing right now. So this pandemic has aroused public interest in the properties, evolution, and emergence of viruses. It is true. Just now, whatever I discussed, a while ago, that where they are present, how they are coming, how this has caused ripples across the world in the technologically and scientifically advanced world. So we are living in a, science and technology revolution era. This, in spite of that, we have faced such a terrible pandemic, which has killed, lakhs of people by now. I'll be showing the statistics also. And under such circumstances, what should the human beings we have to do? What the scientists have to do? What the doctors have to do? So the COVID-19 pandemic exemplifies the constant threat and pressure exerted by viruses on human health and the global economy. We know that terrible loss to the economy. It was a huge loss, to the global economy. It might take, at least 5 to 10 years. That's what the economists are believing or interpreting, that, for recovery, it might take 5 to 10 years for all the countries across the world. The pandemic has triggered an aggressive international response to contain virus spread, cure the disease, and prevent future infections. Nasir et al., 2020. So this is the classification, a brief classification about the viruses, which is popularly called as Baltimore Classification. The scientist Baltimore has given this. Number one group is called as dsDNA viruses, as we have several herpes viruses, adenoviruses, poxviruses. Then single-stranded DNA viruses with positive sense strand, like parvoviruses. Double-strand RNA viruses, like reoviruses. Single-strand RNA viruses with positive strand sense, like coronaviruses. This is where this group comes in. The coronaviruses belong to single-stranded RNA viruses with positive strand. Then we have negative single-stranded RNA viruses like orthomyxoviruses, rhabdoviruses. Then single-stranded RNA orthoviruses, retroviruses, which are with positive strand, and then double-stranded orthoviruses. Uh, these all are the guidelines which are given by ICTV. That's the classification given used presently. Now coming to the coronaviruses. So this is a large family of viruses that usually cause mild to moderate upper respiratory tract illness like the common cold. There are hundreds of coronaviruses, most of which circulate among such animals as pigs, camels, bats, and cats, and of course now human beings. Since the starting of 2002, when SARS coronavirus, that is number one, SARS virus, which has been named as, after the coronavirus number two, which has struck the human population, then it has been named as SARS-CoV-1, the first one in 2002, which has its origin in China, and the second one, of course, also has got its roots in China. So popularly, which is being termed as Wuhan virus. Sometimes those viruses jump to humans, called a spillover event, and can cause disease. So this is the source, like NIAID source I have taken. That means there was a lot of debate which went on, for several days. The source could be the bats or other animals. Still, it is not conclusive that what is the SARS-CoV-2 virus origin. It is going on. The WHO has sent the team. They have not conclusively proved that what could be the pinpointed source. One theory says that it is the lab spillover. Second theory states that it is from the bats. And what is the correct one? Still, we are awaiting. The second team is not being given permission to go and again examine. China has to cooperate with the WHO, and then once the second team goes, and if the China cooperates, then we may come out with the real cause of the infection. Now this is briefly about the viruses and coronaviruses, and the present SARS-CoV-2, the disease which it causes, has been named as COVID-19. Now I would like to throw some light on aerobiology, because my concern here is that for several months it went on like that, that what is the reason, how it is spreading, and how it could spread to the entire world. There's no country which is left without getting caused, which without getting affected by this infection. Now how it is possible, how it is spreading, whether is it spreading by air? And source, of course, the virus has started from the origin place and then has been carried by the human beings only, when they have traveled from the epicenter, the site where actually the epidemic started, or the virus infection started. So branch of biology that studies organic particles such as bacteria, fungal spores, very small insects, pollen grains, viruses, etc. And of late, the studies related to even the materials of inanimate origin or animate origin, which have been included in this field. The importance of airborne nanoparticles, that is NNP standing for natural nanoparticles, or ENP standing for engineered nanoparticles, their study in relation to human, animal, and plant health has picked up momentum as the nanotechnology boom has set in across the globe. A new term has been coined by myself in 2007 conference of aerobiology. I used a term called as nano-aerobiology. So now, even for the viruses, astrobiology has been replaced by astrovirology, which is gaining momentum. Now, I would like to give a reference to these two top scientists, who have contributed a lot. E.C. Stackman and then K.C. Mehta, Indian scientist. He has been, studying, Stockman has studied the plant pathogens, and he studied how they spread, disperse from one region to other region, or from one continent to other continent. And he has been responsible for giving the dissemination of the fungi in case of late blight of potato, downy mildew of and powdery mildew of grapes, and many plant viruses. He spoke a lot. He has done a lot of research on these aspects and has published several papers. And Casey Mehta is known for late Casey Mehta is known for the wheat rust disease, where the fungal rust spores, how they travel. It is a heterocyclic fungus. It has got dual host. One host here on the plains and another host in the hills. And most of us are aware of it. Plant pathology, we have studied about this. And he was responsible for tracking the dissemination of the spores from plains to the hills and back to plains from the hills. And he has established laboratories in the air. That means he collected the air samples in order to prove that the spores can travel in the air and cause infections elsewhere. This is how the dispersal occurs. Selected events of the fungal pathogens, red and blue lines indicating that how they have invaded territories, from one continent to other continent. This can be easily considered as intercontinental transport. Viruses fall under the category of nanoscience and nanobiology by virtue of their size, because the viruses are small sized particles, and because of their nanometer size dimensions, viruses have been considered as naturally occurring nanoparticles. This is given by Nature Nanotechnology in 2020. Now, the viruses range in size of from 10 to 1000 nanometers. Most of us are aware of it. Now, viruses transmitted by air. Is it true that the viruses do not transmit by air? WHO has been stating that the viruses do not transmit by air. In the beginning stages, they said that people should not touch each other. That most of the contagious infections are said to be, spread by touching the infected person by healthy person. So this transmission occurs by contact. That was the wider belief, among us the scientists, that the spread occurs only by contact. WHO was also believing in the same thing, in the beginning stages, that it only spreads by contact. But it took a while for them to, come out of this shell, or come out of this belief that it is not the only way the viruses can spread. If it is so, then people can avoid touching each other, then the infection can be controlled. It would not have become a pandemic. It would not have caused, taken the lives of so many human beings. So therefore, the credence has been given by the American scientists, and then CDC, Center for Disease Control in the United States of America, has come out with the version that the viruses are getting transported by air. Now the question came that how much distance the virus is getting traveled and able to retain its viability and cause the infection. So therefore, I felt that let us go into the details of the transmission of this virus, how it is causing infections, whether air is truly transmitting these viruses. Because when we say that viruses are the obligate ones, they require a living host. But outside the host, once they come out, when the person sneezes or coughs or through the whispers, so then they will survive for some time. How much time? That goes on varying. Its ability, the viral ability, the ability of the virions which have come out. Now, available evidence indicates that a number of viruses that infect humans can be transmitted by the airborne route. So this was given by Couch way back in 1981. He has given to us that airborne route can be used by all these viruses. But he didn't speak about the distance, but certainly all these viruses can spread through airborne route. Smallpox, influenza, chickenpox, adenovirus 4 and 7, measles, Coxsackie A21, rubella, lymphocytic choriomeningitis. All these viruses have been said to be transmitted by air. That means at least to some point, they'll be using the airborne route. maybe passively, not actively. Early studies on airborne spread indicate that viruses and indoor air pollution, has been discussed at length by Koch. Microbiological and epidemiological data have shown that a number of viruses may contaminate the air of rooms and be capable of initiating disease in those who inhale them. That is precisely happened, in this pandemic. That is the reason why the SMS, conditions have been promulgated. The SMS stands for safe distance and masking and then sanitizing. So that is what SMS, message to the people. So that means we must sanitize our hands frequently. We must wear a mask always. Once you are outside the home, you please wear a mask. So that will prevent the infections from one person to other person. What does it mean? That means if a person wears the mask, the exit or entry of the virus will be made difficult. So that is the reason why a mask has been proposed. And but of course, our second wave has taken several lives. One of the reasons could be that people have not followed the COVID-19 rules and regulations properly. The virus as agents of airborne contagion. Let us see how it started. The concept of airborne contagion is ancient, but serious study had its beginning in only 1930s. Further impetus was given in World War II because of problems of respiratory illnesses in military populations. Vernon Knight, who was one of the pioneer scientists, says that in recent years, interest in the problem of airborne viral disease has disappeared, partly due to reduced activity at Fort Detrick, which is the place in United States of America, where a lot of research is being carried out on these topics, and lack of interest of the civilian community in the problem. During the World War II, the greatest contributions and understanding of airborne infection came from the extensive studies of the US Army Chemical Corps biological laboratories in Fort Detrick, Maryland. So, two places, one in Maryland, USA, and then Porton in England. These were two areas where a lot of work has been done. And Vernon Knight has contributed a lot. They have studied the airborne viral infections in 1961. All of them, they came to the conclusion that Wells identified that tuberculosis could be transmitted through air via the nuclei of evaporated respiratory droplets. So the point here is the respiratory droplets. The droplets, once you cough, hundreds and thousands of droplets will come out of your mouth. And these droplets, whether in the wet form or in the dry form, they can dry out also. So those droplets will also carry the virions in the droplets. So then these virions, either in the wet form or in the dry form, will be carried to some distance. So that this is not a new rocket science. This has been known since 1930s. Developed the Wells curve to describe what happens to respiratory droplets after they have been expelled into the air. The Wells curve, which has been named as Wells curve, demonstrates that the respiratory droplets rapidly dry out. Just now I was mentioning about that. Fall to the ground after being exhaled. They may fall in the wet form, or they may dry out and can be carried out by the wind currents, carried forward by the wind currents. How much distance we don't know, because a lot of work has been done by the scientists because of the COVID-19 infection, and there are several hundreds of publications now available, that some saying that it will go to 6 feet and some saying that it may be viable even after 12 feet. And there are some investigations which have mentioned that it can remain viable even after it has crossed the 18 feet limit. In the years up to 1950, William Furthwell summarized a monograph for the Commonwealth Fund. So he was preeminent in developing knowledge of airborne infections. The early studies of airborne transmission dealt almost exclusively with bacterial disease. We know that bacteria, at least we can. The problem with the viruses is that the trapping and identification is a big difficulty. So that's the reason why the scientists do not attempt, because anyone who is interested in doing research, will look for the easy ways. It is a difficult one. There should be methods, there should be instruments, in order to go for the isolation of the viruses and then, studying them in detail. So while the problem of airborne viral disease had to wait the development of virology as a specialized discipline after the World War II. So that's the reason why, after 1940s or 1950s onwards, some impetus has been laid for the research into the viruses. So the role of particle size in transmission, you can see here, the larger sized particles, like we know about the pollen grains and fungal spores, which can travel to several hundreds of kilometers also by causing the infections. And if the larger sized particles which are heavy, if they can be carried out, like PM10 is a particulate matter 10 is the 10 micron size. So most of the fungal spores and then pollen grains fall in this range. They can easily travel with the help of the wind currents. The pollen can cause problems to the human beings in causing allergy, and the fungal spores can damage the crops, can cause infections in the human beings by traveling several hundreds of kilometers. Still, they retain viability. So that means the larger particles, the heavy particles, if they can travel, then smaller particles can easily travel. Bacteria can travel, viruses can travel. And when it comes to viruses, only the problem is, since they are the obligate ones, how far they can go and how much they retain their viability. That is the question which needs to be answered. Because the small particles will remain suspended in air for periods long enough to permit their distribution. 10 micron diameter particles will fall the height of a room in 17 minutes. And 1 to 3 micron diameter particles will remain suspended almost indefinitely, especially if they are periodically elevated by air currents. So that means our viruses fall below this range. So they can easily go, if they are in the aerosol form, and that too in the dry aerosol form. Naturally produced droplets of humans include several types of cells like epithelial cells, electrolytes, and potentially infectious agents like bacteria, fungi, viruses. And they can be carried by breathing, talking, sneezing, coughing. Now, coronavirus family, last 2 decades, let us try to examine this. 3 zoonotic outbreaks caused by coronavirus family. As they are saying, the zoonotic infections from the animals to the human beings. The wilder held belief is that for the first one and the third one, that is the bats, and for the second one, that is camel. SARS-CoV in 2002-2003 origin Fosan, Guangdong, China, and MERS-CoV in 2012 origin Jeddah, Saudi Arabia, Middle East. SARS-CoV in 2019 origin Wuhan, Hubei province, China. So all these 3 are coronaviruses. And SARS-CoV-1, let us try to compare. 842 illnesses, 916 deaths, 29 countries, about 11% mortality. This is from WHO, and CDC. MERS-CoV, Middle East Respiratory Syndrome, 2,519 illnesses, 866 deaths, 27 countries, about 34% mortality. See, the mortality rate is such a huge, but we ignored. The second one is 34% mortality is a very, very huge mortality rate. It has not gone. MERS-CoV is still, now and then the infections are coming. So that means either herd immunity might have set in, only a few people who are susceptible, whose immunity is weak, might be getting this infection and then getting killed. Countries with lab confirmed MERS cases, as you can see here, Arabian Peninsula and outside the Arabian Peninsula. Right? These are the countries, different countries. And summary, this is how infections caused. Just a while ago I have given this. Now coming to the SARS-CoV-2, right? Severe acute respiratory syndrome, coronavirus 2, that is coronavirus infection, viral disease 19. Infections close to 280 million. Deaths, this is as on yesterday evening, above 4.5 million deaths. Just imagine, 45 lakh people died, lakhs. Recovered about 190 billion. Recovery rate is very good, but the death rate is also very high. So many human beings died. Now in India, above 32.7 million people got infected. Above 4.38 million deaths occurred. So that means 4,38,000 deaths. And above 31.9 million people have recovered. People are not getting killed by this infection. The mortality rate is comparatively low in our country as compared to in India as compared to the other countries across the globe. This is about the SARS 2002-2004 SARS outbreak. Countries with confirmed deaths, countries with confirmed infections in red, and then countries without confirmed cases. See that the gray areas where the infection didn't enter during first SARS outbreak. Now after another nearly 20 years, nearly 18 years, the SARS infection has covered the entire globe. During relatively short-lived pandemic, airborne spread of infection on aeroplanes, this has been studied by Yu et al. In 2004 that The same thing happened. Why we didn't take all precautions when we had history of SARS-CoV-1 and then also MERS-CoV, but still we ignored. China could have taken more precautions, in not allowing this pandemic to occur. In apartment buildings in Hong Kong also, they have documented that the aerosol generated by faulty pumping system could spread the virus. And virus in the stool also has been reported in 2020 by airborne transmission. So therefore, the airborne transmission has now become a reality. That is the reason why the people said that everyone must wear a mask, surgical mask, or the mask which can prevent the release of the virions. Influenza H1N1 transmission has occurred by airborne route. This is known. Cowling et al. Have reported it, and most of the viruses, avian influenza, H1N1, all these, we have evidences, a lot of evidence now available that airborne infections do occur. Therefore, people have to take all precautions in preventing the spread of the infections. So this is the case study nature of the epidemiological analysis of influenza A transmission, which has been given by Cowling et al. In 2013. The potential of SARS, MERS, and influenza to spread by airborne droplet nuclei is no longer in doubt, as given by Northel and Nathavitarana in 2020. The reported transmission pathways of SARS-CoV-2 in humans are inhalation by liquid-laden droplets, close contact with surfaces contaminated. All these are possible. And then aerosol transmission has been suggested as an additional yet important pathway for clinical observations in confined spaces. So this is about, again, the airborne route, how the tuberculosis, measles, and chickenpox, which have been studied earlier in different countries and have been established that airborne route is one of the important routes. How it comes is immaterial for us. The infection, whether it is getting caused or not, whether the air is being used as a medium for transport by the viruses or not, passively or actively. Actively, the possibility is less. So it is mostly passively. So whether it comes passively or not, whether it is spreading from one person to other person is there or not, that is to be considered. And therefore, we need to be very, very careful in preventing the further spread of these infections. So this is the global spread of COVID-19 is teaching us how little we know about the SARS-CoV-2 virus. For example, the mechanisms by which that viral disease spreads from person to person and the relative importance of different paths of infections remains uncertain, as given by Jonathan Borak, 2020. We know that all this is happening. And physicist view of spreading. A physicist normally considers that how it spreads by air currents. So with a speed and strength, and it has also caused puzzles to the physicists also, that how it is spreading. And they have conducted several experiments and they have come out with the conclusion that yes, the viruses can also travel through air when the air currents permit. Now, evaporation time of water droplets, just see that 2000 micrometer diameter, it takes 660 seconds, but further 50 micrometer diameter, like in viruses, it takes only 0.4 seconds, for evaporation. That means the viruses which COVID-19 viruses, SARS-CoV-2, if it comes into the air, it can easily dry out within no span of time. That means, it can travel to the longer distance, therefore, thereby adding problem to the human beings, because they don't know, we can't see them, and they will be simply annihilating them if we don't wear the masks. So therefore, all the people, if they wear masks, when they go outside and when they are in the crowds, then best thing we can do is that wear a mask and prevent the infection. In the presence of air resistance, compact heavy objects fall onto the ground quickly, but the lighter ones can travel as exhibited by the Brownian motion and follow the pattern of turbulent convection, and therefore they can easily travel from one place to other place. The transmission of COVID-19 disease, however, still depends on the infectious virus load carried by the droplets, and therefore people coughing, sneezing, breathing also releases. So they need to be very careful, follow the SMS guideline, then it will be easy for us to prevent this particular infection. So they can float in the air up to 10 minutes, as done by MIT, Massachusetts Institute of Technology. The scientists have come out with this observation. So this can be inhaled, and therefore they will also follow the patterns, and therefore the later is determined by many factors such as the location of doors and windows, ventilators, and therefore doctors and scientists are suggesting that when the patient is inside, when the persons infected people are inside the homes, please open the windows, have enough ventilation so that the viral particles, even if they are released, they will just go away into the outer air. So thereby we can at least reduce the infections. So this is the WHO scientific brief as given on 29th March 2020, that transmission also occurs by the air route. So ultrafine particles and the lungs. This is an established fact that the viruses fall under the ultrafine particles UFP, as they are below 1 micron size. Therefore, this is known and established, and why we should keep away the viruses from these ultrafine particles, because either inanimate or animate, once they are in the outside atmosphere, based on their size and based on their aerodynamic diameter, they can travel to any distance. So important factors characterizing airborne SARS-CoV-2 transmission remain unknown. So therefore, we are just in the beginning stages of the pandemic. The only 1.5 years we have gone through, and I think we have to be very, very careful with this until the time all the people get vaccinated, and all the people wear the masks and avoid the virus from spreading from person to person. New review explores effective sampling techniques for collecting airborne viruses, and those interested people can follow this. There is a need now for the scientists to come out with the isolation and identification of these viruses by different methods. This is the recent paper by Prashant Kumar, published this year only, and they have established some air sampling methods discovered, and they are using that successfully. So this is about Prashant Kumar's paper and their interpretation that how it could spread. The common central air conditioning system can spread the viruses very effectively. We know that China has come out with these examples early. So let us vaccinate the people. Aerobiologists must play a proactive role in facing challenges posed by zoonotic infections, and we must also keep in mind that we need to live with viruses. Therefore, the herd immunity is one of the sources where we must achieve, and which may take a while. So therefore, let us all together fight this particular infection and come out of this dreaded pandemic, which has no solution as of now, even complete solution. I mean to say that we need to do the vaccination, complete vaccination, and then secondly maybe booster doses, and then secondly the avoidance, that is by using SMS principle, sanitization, and then safe distance, and then also masking. Thank you very much. Dhanyavad for your patient listening.
So I am very thankful to you, sir. Dr. Rao sir has given very nice lecture, wonderful lecture, informative lecture, and he has described the different types of viruses along with COVID-19 virus. And for the first time, I came to know that the viruses are also transmitted through the air, because before this I was under the impression that viruses are contagious and they can spread, or they can be transmitted only with the help of touch or other things, with the help of vectors. But today I came to know that viruses are also transmitted through the air. So I am very thankful to you for sparing your valuable time, and I call Dr. Bharate sir for the vote of thanks. So over to Dr. Bharate sir.
On the behalf of Dadapatil Rajale, Arts, Science and Commerce College, Adinathnagar, Department of Botany and Internal Quality Assurance Cell, organizes the one-day national webinar. I express my sincere thanks to Dr. K.S. Ramchander, Consultant Aerobiology, Director of Abu Hussam, Muscat, Sultanate of Omen. Sir has delivered lecture on COVID-19, basic understanding and aerobiological perspective. During his lecture, he has described viruses, bacterioviruses, pathogens from outer space, the need to redefine viruses, classification of viruses, Baltimore classifications, coronaviruses, viruses transmitted by airborne route, contribution of William Humwell, coronavirus family, SARS-CoV-1, ultrafine particles and lungs. I express my gratitude to Honorable Appasaheb Rajale, President of the institution, Honorable MLA Munikatai Rajale, Shevgav Pathardi, Honorable J.R. Pawar, Secretary, Honorable Shivaji Rao Rajale, Chairman, CDC, Honorable Rahul Rajale, Trustee of DPR College. I express my gratitude to Principal Dr. R.J. Temkar, Professor R.T. Gholap, IQAC Co-ordinator Dr. J.M. Nehul, Head Department of Botany and in charge of Science Faculty, Dr. M.S. Tamboli, in charge of Arts Faculty, Dr. H.J. Deshmukh, in charge of Commerce Faculty, Professor C.N. Mansare, in charge of Computer Science, Professor Rohit Adlim, Dr. Atul Sorpagar, Head Department of Zoology, Dr. S.D. Muscat, Head Department of Chemistry, and all the teaching and non-teaching staff of DPR College, Adinathnagar. With kind permission of Principal, I conclude the national webinar. And thank you, sir.
So thank you, sir, for vote of thanks. And as the head of the Department of Botany, I would like to express my deep gratitude towards all the participants for joining this national webinar on COVID-19, basic understanding and aerobiological perspective. Thank you very much. Thanks.